Friends, today in this lecture we'll discuss about Western blotting. So Western blotting is an important technique that is used in cell and molecular biology to identify specific protein from a complex mixture of protein extracted from cells. Okay, so the major purpose of using this process is to identify a particular protein, our target protein that we want to see whether it is present in a cell or not. So let us discuss. So it is an immunoblotting technique that rely on the specificity of binding between the molecule of interest and a probe to allow detection of our molecule of interest in a mixture of many other similar molecules. So this technique basically relies on the specificity. For example, when we will extract any protein from the cell, apart from our target protein, there will be many proteins which will be present there. So we have to add a particular probe that will go and bind to our target protein only and not other protein. So this technique rely on this interaction mainly to see whether our particular protein is present or not. So so the probe generally used here is antibody, and our target protein or the molecule of interest here is generally our protein which we want to see whether it is present or not. So let us discuss about the procedure of this Western blot. So the first step in this Western blotting experiment is to lyse the cell in order to extract protein, because we need to open or lyse our cell so that we can isolate the protein. Okay, so we have to lyse the cell. So in that case we use detergent so that cell can be lysed and all the proteins can be extracted, which in which our target protein will be present. So protein extracted here are then mixed with gel loading dye, which usually consists of glycerol, uh, beta mercaptan, SDS. All these are present in gel loading dye, and our extracted protein are mixed with them, and they are run on this SDS page gel. Now this SDS page is widely used for protein separation. For our DNA and RNA, we are using agarose gel. For DNA and RNA, we are using your agarose gel, because the size of the pore of this protein of this SDS page is usually very small, and protein being very small in size can easily pass through this gel, while DNA RNA being very large in size will not be able to pass through this. So we mostly use agarose for DNA RNA, because the size of the pore of the agarose is generally bigger compared to your SDS page, and the pore of this SDS page is smaller, so protein can easily pass through it because the size of the protein is very small compared to DNA and RNA. Okay, so these pages can also be used for very small size of DNA, but for protein it is mainly used. So so the proteins here will be separated based on their size. So larger protein will remain at the top, okay, while smaller protein will migrate faster because they will uh, encounter less resistance compared to this large size protein. So the small size protein will move faster, okay, and the large size protein will move very slow because they will encounter huge resistance. So this protein are separated here. All the protein that has been extracted from cells are separated here. Next, these proteins are transferred to your membrane. Now we have to keep in mind that the protein being carrying the negative charge will move always towards the positive pole. So we will keep our gel towards the negative electrode, because our protein will pass from negative to positive towards the positive electrode. So in order to pass the protein from gel to membrane, we keep the membrane towards the positive pole, and we keep the gel towards negative electrode, so that protein can move from this negative to positive side. So this is one of the major step in this process. This the placement of this membrane and gel is one of the major step that everyone needs to know, where these two things should be placed. The gel should be placed towards your negative electrode, and the membrane should be placed towards positive electrode. Okay, now after the protein has been passed from this gel to membrane, the membranes are usually soaked or kept in BSA or dry milk, which usually contains no fat, because uh, we do not want the antibody to bind non-specifically in the membrane. So addition of this BSA and milk casein will block all those non-specific site, so that our antibody or the probe which we will use later upon addition will only bind to our target protein if it is present there. Okay, it should not bind to any other protein in any other site in the membrane. So to avoid those things, we usually block the membrane with BSA. BSA is widely used because sometime in milk our casein and other phosphoproteins are also present. So BSA is widely preferred. Okay. So after the blocking is done, we add a primary antibody. This primary antibody is usually a monoclonal antibody, which is very specific to our target protein. It will only bind to our target protein. It will not bind to any other protein. This antibody is very specific, so it will go and bind to the target protein if it's present there. Okay, we can see here it has been bind to our target protein. After that we will wash it, so that any unbound antibody will washed away. It will be washed away, and only antibody that has been bind to the protein will be retained. Okay, next we will then add secondary antibody, usually known as labeled antibody, because it has been labeled with an enzyme which will cleave a substrate to give a color. So first we will add the secondary antibody. The secondary antibody will go and bind to the Fc region of our primary antibody. Okay, now when this will go and bind to the Fc region of our primary antibody, we will again wash away, so that any unbound secondary antibody should get washed out, and we will then add our substrate. Okay, now the enzyme that has been linked to the secondary antibody will cleave this substrate to produce color. A color will be produced. Okay, now this color can easily be detected by X-ray film. So this color will only be produced at those sites where our desired protein is present. So if our protein is present, we will get a color in the membrane, and we will get to know that yes, our target protein is present or not. So only those places in the membrane will give out color where our target protein is present, and we can see that yes, our target is protein is present if we get any signal, and if we don't get any signal, that means there is no target protein is present. So I hope you guys have understood the entire Western blotting, and thank you for watching. If you like it, please like and subscribe. Thank you.